Okay, here we go. So welcome everybody to week 2. This is the first lecture of the week. Today we're going to be talking a lot about population and individual health risks. Essentially today's lecture is really meant to help you guys with your assignment 1. So for those of you attending, this will be particularly useful because you'll also spend today and tomorrow's tutorials talking about assignment 1. So hopefully by the end of this week, everything will be crystal clear and you guys can get started on that assignment right away. So what were the 3 main study types or the study designs that we covered last week? Does anyone want to refresh our memory how we ended things last week? Anybody remember what we talked about? Dailies, Kayleys, Haleys. So those were health indicators that we talked about. But does anyone remember the designs? Descriptive, experimental, observational. Yep, exactly. So we have them there. Now today I'm going to work a little bit on comparing those study designs. Again, for the context of assignment 1, you will mostly be forced to explore observational experimental studies, not so much descriptive, but it's always good to know what all of these are and how they compare to one another. I will also say that for those of you that remember last week, we also talked about observational studies in the context of 4 subtypes. So those were ecological, cross-sectional, case control, and cohort studies. So those are the 4 types of observational designs that we covered. Now, how do these study designs compare? Most studies in health geography do measure association between some exposure and some health outcome, and most of them will be observational. Again, we talked about this towards the end of the last lecture, why it's quite rare to see experimental designs in the context of health geography, especially using humans. It's much more common that you'll find animal studies instead, or eventually you might find research that's related to pharmaceuticals and random control trials that are testing the efficacy of drugs, but it's not as common in terms of epidemiology or health geography to find those types of studies because we generally try to avoid using humans as study subjects, particularly for any hazardous substances or exposures that we're not quite sure what the full results might be in the body, how they might impact someone's health. Now, the reason I talk about this is because we're going to spend some time talking about these different studies, but also how they rank compared to one another, how their results and how their associations tell us different things. We also mentioned this a little bit last week, how some designs can give us an indication of causality, and we'll break that down a little bit more today. So this is to say that the ranking of these design types is really important because the findings that you might get from different studies, from different study designs, they're not all weighed the same, right? We might want to weigh the results from one particular type of study a little bit higher compared to another. So we'll talk about that. There are also limitations associated with each type of study design. And again, we'll kind of break those down by the different design types. So to start us off here, this is a handy little ranking scheme that I found in that article. And so it breaks it down between the observational and the experimental types. So usually we would weigh the experimental designs quite a bit higher in terms of their methodological rigor compared to observational types. But within the observational types, we talked about those different subtypes. So again, cohorts would usually be weighed as a little bit more stringent, a little bit more rigorous compared to something like case control or a cross-sectional survey. Now, what do all these study designs have in common? Well, they all try to characterize some sort of association between an exposure and a health outcome, or some kind of environmental, social, whatever risk factor determinant of health. Now, in order to understand the relationship between those things, you need to know how they're associated to one another. You need to know how to characterize that relationship between those variables, the exposure variable and the health outcome. So we'll talk a little bit about that. So measures of association, again, are just those relationships between two or sometimes more variables. An example of a type of association is a correlation, which many of you have probably heard or come across in some academic research or health studies. Now, we often try to characterize the association in order to understand it better, in order to understand how the two variables relate to one another. So one way that we might characterize an association would be based on the direction. So when we're talking about the direction of an association, we either mean that it's positive or negative. If it's a positive association, then we're talking about an increase in one variable leading to an increase in the other variable. Contrarily, if we're talking about a negative association, then that means that the direction is opposite. So an increase in one variable actually leads to the decrease in the other variable. Another way that we might try to characterize a relationship between two variables is whether or not it's strong or weak. So whether the strength of the relationship is in one way or another. Now, if it's a strong relationship, generally there's less variation between the values. If there's a weak relationship, then we say that there's more variation between the values. So you can kind of see the figure there that I've added just to show you how these might visualize if you were to plot two variables against one another. Now, this is to say that association does not equal causation, and we'll unpack that a little bit next. So you guys have probably heard this before. Causation is not correlation, or correlation is not causation. And that's because the associations between variables can be causal or correlation based, or they can have a completely different kind of relationship that we won't get into in this course. But so an association is just, in plain language, a relationship between two variables, whereas causation implies that one variable causes the other. Now, to illustrate this, I put an interesting graph here that I found, and it's showing the number of people who drowned by falling into a pool versus the films that Nicolas Cage appeared in. So these are two variables that probably don't have anything to do together, but when you plot them, they might actually look like they're correlated. So this is what we would call a spurious correlation, right? It's a little bit dubious. It's not telling us anything about the association or the potential causal relationship between the two variables, because likely there is none. But a correlation is still technically a type of association. It just tells us that there's some relationship there, but it's not really able to tell us the direction of it or the strength of it. It doesn't really tell us about whether or not the relationship is actually true. So something to keep in mind whenever you're reading a health study and you're looking at correlations or associations, you know, it's helpful to try to characterize them and try to understand how much meaning you should ascribe to the particular relationship between two variables. So in health geography and epidemiology, we're usually focused on exploring the relationship between two very specific types of variables. Again, this is the exposure variable and then the outcome variable. But to establish causality, or to prove that one causes the other, you need three different things. You need three sort of criteria in order to establish causality. Now, the first one is association. So we just talked about that. It's the relationship. But specifically, it needs to be a relationship that's causal. So a change in the exposure, it needs to cause some change in the probability of the outcome, whatever that health outcome might be. The second criteria that you need in order to have a causal relationship is that you need some aspect of time or temporality. So the exposure to that whatever it is, a substance, a risk factor of some sort, it has to come before the outcome Y. Now, the third criteria is direction. And again, we talked a little bit about this, but essentially you need to know that it's the factor X that's causing the outcome Y. It's not the other way around. So the direction is only one way. If it was multiple ways, then it would be a very confusing relationship. You wouldn't be able to know whether this was in fact a causal relationship between exposure and outcome, or whether this was some sort of confounding variable. So we won't get into that. So for the simplicity of this course, anytime that we're talking about causality, it means that we need to establish those three criteria: association, temporality, and direction, in order for it to be a causal relationship. But as you might imagine, it's quite difficult proving definitively that something causes another thing, especially in health research. In fact, it's much more likely that you'll encounter health studies that say that some exposure increases the risk of some health outcome. So what is a risk? What do we mean by health risk? Well, generally it's defined as the chance or the likelihood, probability that something will happen to your health, that something specific, some exposure will impact your health. Now, as we learned last week, when we talked about this concept of a socio-ecological model, right in the figure there, many different factors can influence your health. There are many different risk factors. So that can start at the individual level, but those factors can branch out all the way to global scales, ecosystems, environments, things like that. So this makes it really difficult to characterize an association between two variables, and it's not always super simple. It's not always really straightforward. Instead, you might find health studies that talk about the presence of some variables increasing the health risk of that outcome. So for example, we brought this up last week as well in lecture. There was a study that talked about proximity to highways and the risk of asthma. So that might be one example there of them trying to characterize an association between exposure and health outcome without necessarily being able to establish that causality. So this essentially leads us to this conclusion that disease etiology, which is the study of causes of diseases, it's a really complicated field of research. It's not as straightforward as saying, exposure to this causes outcome that. It's in fact a lot more complicated than that. And this is because a single disease can have many different causes, many different risk factors, many different determinants, as we also call them. How's my pace, everybody? Are we following along pretty well? Am I going a little bit too fast? Feel free to interrupt me. Pace is good. Okay. Yeah, if any time you find that I'm rushing through things, do let me know. I want to make sure that everything is clear, especially because this will really help you guys out for your assignment. Okay. So proving causality is even harder when the health effects from that exposure don't occur right after that exposure. So what I mean by that is when we're thinking about something like an acute disease versus a chronic disease, how would we compare those? Well, generally an acute disease has some sort of rapid onset of symptoms, right? You're exposed to something and then within minutes or hours, you have symptoms from that exposure. That means that the disease generally is pretty quick to observe, and you're much more able to link that exposure or that geographic phenomena to that exposure or to that health outcome because it's still there. It only happened, let's say, half an hour ago. Instead, with chronic diseases, as you can imagine, that's quite a bit more challenging to do. The symptoms sometimes don't happen for months, years, decades. Many chronic diseases progress over a really, really long period of time. And so it's much more difficult to link that exposure or that geographic phenomena to the particular health outcome that you're now witnessing. So how does this impact causality? Well, we talked about this a little bit last week, this concept of disease latency, right? So this is the time that it takes for a disease to sort of present itself after the original exposure that took place. So that's disease latency. It's that period of time. Basically, it's that little kind of question mark that you see here in the figure. So this impacts causality because it impacts temporality, right? If you don't know for sure that exposure came before the outcome because maybe that exposure happened so long ago, you have a lot of challenges trying to establish whether or not it's a causal association or whether there's causality in that relationship. So that's something to keep in mind. Now, if we read a health study that found the association, it found temporality, and it found the direction, right? That exposure caused outcome. Can we say that the exposure therefore causes a health outcome of interest? What do you guys think? Is that enough evidence to suggest a causal association? Yeah, I can repeat that. So if we read a health study that found association, it found temporality, and it found direction, right? We said that those were the three sort of main criteria to look for if you're trying to establish a causal relationship between variable exposure and the variable outcome. So if you did find those three things, is that enough evidence to suggest a causal relationship between that exposure and that outcome? No, it's not enough evidence. Why not? We said that that's all you needed. So what else do you guys think? Why are we saying no? Anyone want to elaborate? You can feel free to explain over audio or chat, whatever you feel more comfortable with. Could be multiple causalities. Yeah, if the study hasn't necessarily adjusted for confounding variables, for example, yeah, that could definitely impact whether or not you could say for sure it was a causal relationship. What else? Why have some of you guys said no? What else would you need to see? Anyone else want to add a response? Would you need to see a trend? Interesting response, Zoe. Yeah, so that does sort of bring us to the correct answer that I was aiming for. It would suggest causality, right, if you were able to meet those three criteria. But in order to know for sure, you're right, Zoe, we need to see a trend. We need to read more studies in order to know for sure whether this was just a fluke in that one particular health study or whether other studies are also finding this potential causal link. So you're absolutely right. We need to see some sort of trend. We need to know if there's other studies that are also looking at this. So this brings us to the very fun part of today's lecture, where we talk about the Bradford-Hill criteria. Now, the Bradford-Hill criteria is part of the reading that I assigned for this week. So not sure whether you guys did the reading yet, but if you haven't yet, I absolutely would recommend doing it before you get started on your assignment one. It's basically a recipe book for how to do assignment one. So you'll find it very, very useful if you haven't already given it a look. But we will break it down today in the lecture. So it's called How to Read Clinical Journals to Determine Causation. It's an article by the Canadian Medical Association, and it was published back in 1981. Yes, I know it's a very old article. Full disclosure, I was actually assigned an assignment like this back when I was doing my undergrad in geography. And I was so kind of taken aback by how useful this particular type of assignment was to apply the Bradford-Hill criteria that I thought, oh, I'm going to make other students do it. No, but honestly, I think you will find it to be a pretty useful assignment to do, hopefully when you reflect back on it in time. Because essentially it forces you to apply these really simple principles, these set of criteria in order to know whether an article is showing causation or whether that relationship that the articles are talking about are likely to show some sort of causal link. So essentially the Bradford-Hill criteria are a series of criteria that you apply to research studies, and it's helpful for you to get a sense of whether some exposure that you're interested in is associated to that health outcome. And it also kind of teaches you how to assess different strengths of associations, especially when they're all from different studies. So how to actually compare different study designs against one another in order to help you kind of understand what the relationship is between that exposure and that outcome. So the Bradford-Hill criteria are a very, very useful thing to know. Now, the 9 criteria can be sort of grouped into these three broad categories, right? So 3 of the criteria really have to do with the association between the two variables, between the exposure and the health outcome variable. The other 3 have more so to do with comparing it to other studies, other evidence. And then the third group, there's 3 criteria that have to do more so with the specific exposure outcome relationship. And we'll talk about each of these 9 criteria individually. So get comfortable, get cozy. We're gonna go through this whole thing. So step 1 of the Bradford-Hill criteria is where you identify the study design. So these are the study designs that we've talked about so far in the course. Granted, there's a lot more study designs out there that we haven't covered, but for the purpose of assignment 1, for the purpose of this course, we're going to keep things pretty simple. So again, what were they? Well, we talked about experimental designs, and then we talked about observational. And within the observational category, we talked about 4 different types. So this was the cohort, the case control, the cross-sectional, and here they call it a case report because I took this diagram from something that was specifically talking about clinical health studies, so more in medicine. But let's say the ecological design could be roughly equivalent to a case report. So we would generally place it underneath the cross-sectional survey when we're trying to weigh the evidence from those different studies. So that first step is just identifying what that particular health article, what their methodology was, what kind of study design they used, and assessing whether or not it's generally a weak or a strong method. Now, technically, only experimental randomized control trials can establish that causality, right? Because they're the only types of study designs that can meet those 3 criteria really easily. But as we also kind of touched on last week, cohorts technically can also help you establish that causality because unlike the other observational designs, they do establish that temporality. Remember that this is because the cohort follows a group of participants over the course of time. So they enlist people in their study and then they follow them over the course of many years, sometimes decades, before they get that health outcome that you're interested in studying. So for the purpose of this assignment, we will say that experimental designs, which can include human and animal experiments, as well as cohorts, generally you're able to tell something about a causal relationship from those 2 types. Hopefully that's clear. Please let me know if you want me to repeat any of that or if any of that seemed unclear. Are we good to move ahead? Can you just repeat the last part? Yes. What was I just saying? So I think I was talking about the RCTs and the cohorts. So to establish a causal trend, right, we need those 3 criteria. Now, all of the studies will be able to tell you something about the association part, so we know that that's fine, but it's the temporality criteria that's particularly hard to meet in most epidemiological or most health studies. So that's why only the experimental designs and only cohorts are really able to tell you about that causal link, that causal relationship, because experimental designs and cohorts are able to tell you something about temporality, right? Especially in a cohort, they're following participants over time. And so you know that that exposure came before the health outcome because you were able to follow those participants over many years, sometimes decades. Does that sound better? Is that more clear? What about RCTs? Well, glad you asked because we're going to talk about that next. So the next step of the criteria, so after you characterize what kind of study design it is, the next step is to ask yourself, is there evidence from experiments? Now, why is this so important? It's because if it's not an RCT, if it's not some sort of experimental randomization, then you can't definitively say that that health outcome is particularly linked to that exposure without the potential for confounding variables. Now, why do we say this? It's because the random assignment in experiments is such an important part of that type of methodology. So that's what makes a randomized control trial or any sort of random experimental design really, really strong. It's because of that random assignment. Think about it from this perspective, and I kind of have it shown there in the figure. If you are able to divide up the group of, let's say, patients or participants, we'll call them, if you're able to randomly assign them a treatment or an exposure, and you have that control group that wasn't exposed to whatever that you're studying, when you follow them up, you know for sure that in the group that had the treatment, the exposure was the thing that caused the health outcome. It wasn't any other variable because you've randomly assigned them. You've controlled for those other types of potential variables by doing that random assignment. So that is what makes the experimental design so strong. And that's why it's really the best type of study design if you're thinking about it from the perspective of being able to distinguish whether it's a causal relationship. So because of that, we say that these types of designs have really, really high internal validity, right? Because the methodological rigor is just so strong with any sort of experimental random design. I think that's all I need to say here. Oh, contrary to internal validity, when we talk about external validity, which is how relatable the outcome of that study is compared to other studies, do you guys think that an experimental design like this would have high external validity? What do you guys think? Reed says no. Why do we think no? Anyone want to elaborate? Too many controlled variables. Okay, I think I see your logic there. So because it's such a controlled experiment, it's not necessarily relatable to the real world. I think that's what you're getting at. Yeah. Yeah. Anyone else want to offer an idea why an experimental design might not have very high external validity? If it's a causal relationship, then it should have high external validity with other similar studies, right? Well, it depends on the study. So let's break this down a little bit. If I have an experimental design that uses animals, do you guys think that that would have very high external validity for something that we're interested in studying for humans? But can you generalize it? No. Yeah, so exactly. It's a little bit subjective, you could say, right? How much validity a particular study design has. And by the way, there's no right or wrong answer per se. It's really how you explain it or how you justify it. So even though we know that an experimental randomized design has really high internal validity, right? We know that you're controlling for lots of things, therefore it has a really, really strong methodology. Generally, we would say that the external validity isn't that high because you can't really generalize it to a real life scenario. In real life, exposures are really messy. You're often exposed to multiple things that you can't really address. You're not also testing sometimes relationships that are interconnected. So sometimes multiple variables need to be present in order for a particular health outcome to happen, and you're not always looking at that. So this is to say that generally we would say that an experimental design does not have particularly high external validity, but it does have high internal validity. So hopefully that difference there is clear. Now, the next step is where you apply those 9 criteria. So again, I'm going to go through each of the criteria individually. Hopefully we have enough time to get through all 9 of them. So the first criteria out of the 9 is whether the association is strong. This sounds pretty simple, and yet when you're looking at different studies that are using a bunch of different methods and are using a bunch of different health indicators, it can actually get pretty messy and a little bit confusing to compare them. So we're going to try to simplify this as much as possible so that it's really clear and easy to follow along. So studies have a lot of different measures of association, right? We talked about one example is a correlation. And one way to show whether something is correlated is something called a Pearson's R. I'm not going to spend a whole lot of time talking about that because, hint, hint, it does not appear in your assignment, or at least it's not a critical part of your assignment. So you don't really need to know that in a whole lot of detail. And I'll save that for whatever stats course you have to take over the course of your undergrad. But something that you do need to know is statistical significance, because a lot of the health studies you might encounter say that they'll give you a set of results, but they'll tell you that there is an association that was statistically significant. And what does that signal to you? Well, it signals that that's probably the most rigorous result that they're citing, because it's the one that passed whatever statistical threshold that they set. And what it means is that they're confident in that value. They're confident that that particular result wasn't due to some sort of chance factor. It's because of the relationship that they actually have that they're summarizing. Now, strong associations between exposure and outcome will usually cite a very small p-value. But what's really common to encounter in health studies, especially cohorts or case controls, is something that we call a relative risk or an odds ratio. Sometimes this might also be called a hazard ratio. And these are all very similar types of measures for that relationship between the exposure and the outcome. So you might find in a study that they say exposure X and outcome Y, the relationship between them had a relative risk equal to 1. How would you interpret that? Well, you would say that the exposed and the unexposed groups, right, that they were comparing were equally likely to get the disease. In other words, there was no association between the exposure and the health outcome. So that's if a relative risk, odds ratio, hazard ratio, whatever similar measure equals 1. If they equal to some number smaller than 1, so usually between 0 and 0.99, let's say, what that means is that the exposed group is less likely to get the disease than the unexposed group. In other words, the exposure actually had a protective effect, right? It resulted in less disease than the people who were not exposed to whatever it is. And you might see that kind of thing in some sort of health intervention study, right? Where they're trying to see whether some new device, for example, saves lives, then you might see a relative risk or an odds ratio that's smaller than 1, because they're seeing whether or not it improves the health outcome, not whether it causes more disease. Now, you might also encounter a study where a relative risk or an odds ratio is equal to greater than 1, right? It's bigger than 1. That means that the exposed group is more likely to get the disease than the unexposed group. In other words, there's a positive association. And whether or not it's a strong association depends on what the actual value is. So the more that it's bigger than the 1, the more strong that relationship is. If it's a value that's, let's say, just a little bit above 1, then you would say that it's probably quite weak. Now, let's say I told you that a study gave you a relative risk that was higher than 1, but that they said that it was not statistically significant. How would you interpret that association? What would you say about it? So even though it's greater than 1, they say that it's not statistically significant. What do you guys think? How would you interpret that? Any ideas? Would it be a correlation at that point? Interesting. Would you disregard it? Yeah, so you guys all have correct answers. You're all thinking about this the right way. It's more likely due to chance. Yeah. Basically, all that statistical significance is guaranteeing you is that it's probably not due to chance. So if you don't meet that threshold for statistical significance, it doesn't say anything about your association per se. It just means that it's not statistically significant. Therefore, you can't rule out that you got that particular value due to chance. That's all it means. How you interpret that is that, yes, you might say that there is still an association, but maybe you would have to do it, the experiment more times. Maybe you would have to run the statistical test more times to see if it's continuously not statistically significant. It might be due to the fact that you haven't adjusted for some kind of factors, some kind of confounding variables, that it's giving you kind of a wonky association. So there's many reasons why you might not actually have that statistical significance. It might also just be that you haven't had enough study subjects in your experiment, right? So there's many reasons why you might not meet that statistical significance threshold, but it doesn't mean that the association is garbage. It just means that maybe you would interpret it with quite a bit of caution, right? You wouldn't want to make any sort of big policy decisions based on an association that did not have that statistical significance. So that's just something to keep in mind, because I don't know, you might encounter some of those in your assignment. I couldn't say for sure. Now, I do want to touch on confidence intervals, because you are also likely to encounter confidence intervals in your assignments. So confidence interval is just sort of a series of values that are meant to serve as sort of an envelope around that value. So all of that it tells you is that you're confident that the value that they're citing, so let's say it was a relative risk of 1, and they provide you some sort of kind of envelope of values to go around it, so that you know that the true value is likely to be in that range of values that they've provided from the confidence interval. If you really wanted to get into the nitty gritty, it's because most of the time it's a 95% confidence interval. And so all that it's telling you is that 95 out of 100 times that you would do that test, you would be likely to get the value within that range of values. But this is not a statistical course, so you don't need to really know the nitty gritty of that. But in case you're curious, that's why. But for the purpose of this course, a confidence interval essentially just means that it's a range of values that allow you to be confident that the association is within sort of that range of values. So I'm going to present you with another hypothetical scenario. Let's say you have a relative risk of 1.1, but the confidence interval that the health study cites is between 0.7 and 1.3. Now, I bring this up because we know, based on that table there, that 0.7 means that it's protective, right? It's smaller than 1. And yet on the other range of the confidence interval, it's greater than 1. It's 1.3. So if it falls between those two ranges, how would you interpret that? Any ideas? We ignore it. It's null. Do people agree with that? Anyone else have an opinion? So again, there's not really a right or wrong answer to this. If the calculated relative risk is 1.1. Yeah, so you bring up a good point. If the value that you got was 1.1, but the confidence interval was quite large, you agree with Elena. Yeah, so if you were to use this health study to, again, make some sort of policy decision, right? Like, let's say you're going to ban a substance because one study found a relative risk of 1.1 and it might signal a relationship. It depends on what the consequences of that decision would be, right? So if you're trying to find an association because you are worried about some substance causing harm in a population, maybe you would still be confident enough in that kind of relationship because you might think that the consequences of ignoring it would be quite a bit worse. If, on the other hand, you were looking at this association and it wasn't a life or death scenario, maybe you would say, not that confident. The confidence interval is too wide. Therefore, the association's probably weak. It might not actually be true. Maybe it was some sort of fluke in this particular study. So it really depends on what you're using the particular study for. But as a rule of thumb, yeah, generally you would not be overly confident in that kind of relationship. You would probably want to look at other studies and see whether this was consistent or whether other studies found other relationships that are maybe stronger, a little bit more rigorous. So hopefully that's clear. There's a lot of nuance to this, in other words. So, don't be scared to sometimes interpret things that are not crystal clear, that are not black and white. In the real world, it's quite likely that you will encounter these types of relationships pretty often. So you don't have to be scared of them. There's no right or wrong answer per se. Okay, so the third criteria is whether the association is consistent. And so we touched a little bit on that from the previous slides, because the more that you see a relationship consistently, the more confidence you can have in it, right? The more the reliability will be high. This also tells you that there's reproducibility from the associations, because if many different studies are finding a consistent relationship, even though they might be using slightly different methods, then that just means that the answer or that result that they all have is quite reproducible. And that's always a good thing. The next criteria is that temporal relationship that we're talking about. So again, this is really a crucial element of health geography studies because it's so important to establish whether or not the exposure came before the health outcome. And as we've talked about, it's pretty hard to do unless you have particular types of study designs. So it's very, very difficult to establish this, especially for diseases that have that long latency that might take several years or decades to manifest themselves in somebody. So as an example there in the figure, it's plotting the number of cigarettes smoked per capita, and it's also plotting the lung cancer mortality over time. Now, you'll notice that there's a difference of a couple of decades there. There's that time lag, and that's again because of the latency. So because it took many decades for people who were smoking to actually develop lung cancer. This is why when they did a lot of studies originally on the relationship between cigarettes and lung cancer, they weren't able to establish that causal association right away because it took so long. They weren't confident because of that temporal lag. So they had to wait for many experimental studies to happen, especially animal studies, which really definitively told us for sure that cigarettes do in fact lead to a significant increased risk in lung cancer. Now, the next criteria is the dose response. And so the dose, as we know, just refers to the quantity or the frequency of that exposure. So it depends on the hazard type. If it's something like a substance, then you're usually talking about the weight or the volume of that particular exposure. But for something like radio frequency, maybe you would talk about the frequency of exposure. So it's a different type of way to measure dose because it's a very different type of substance. Now, usually a dose response is a fairly simple kind of relationship where an increase in the dose, right, an increase in the amount of exposure generally leads to an increased response or a health outcome. In the real world, these are not always simple linear relationships. And so for example, there I've got the figure of blood lead quantities in IQ. So you see there that as you have more lead in your blood levels, the quantity of lead per blood, the IQ levels decrease. So what kind of relationship is this? Is this positive or negative? Negative. Yes, correct. Great. Now, the next criteria is whether the association makes epidemiologic sense. What does that mean? Well, essentially, this is where you have to compare it to other studies, other epidemiological studies, and specifically whether or not the association was also found in other geographic areas, whether it was also found in different populations, maybe similar populations. So an example of this is when they were studying whether the use of Roundup, which is a type of herbicide, so whether Roundup use among farmers led to an increase in cancer cases in those farmers. Now, they had some studies that were done in places like North America that showed a potential association there, but it wasn't until they gathered more studies from places like Europe, from other countries, in the same type of relationship was found consistently between different farmers in different countries in different epidemiological studies that they were able to say, okay, this is probably a stronger relationship. The association is probably strong because of that. The seventh criteria is whether it makes biological sense. So essentially, it just means whether the relationship is biologically plausible based on what you already know about the human body, the way that cells, tissues, organs, systems work. An example of that is when people were studying the effects of cigarettes again, they knew that the chemicals in cigarettes tend to bind to DNA. And once they're inhaled, they particularly result in impacts in the lungs. Right. So because of this biologically plausible relationship that inhalation obviously would impact the lungs, they were able to say, again, more definitively that cigarettes cause lung cancer. The next criteria is whether the association is specific. Now, this rarely happens. So it's quite rare that you'll actually be able to meet this particular criteria, but occasionally it does. And that's because the health outcomes that you're generally looking at have many different causes. It's really, really quite rare to find a health outcome that's linked to one specific exposure. One time that this did happen, that's a pretty well-known example, especially in environmental and occupational health, is mesothelioma, which is a type of cancer that happens on the lining of the lungs. Now, it's particularly noteworthy because there's really only one established cause, and that's asbestos exposure. And the reason that they found this cancer was because it was so predominant or prevalent in populations of miners who specifically worked in mines for asbestos. So that's one example of a specific association, but in all honesty, you will probably rarely be able to meet this particular criteria. Now, this brings us to the ninth and last criteria. And perfect, we still have a couple of minutes left. So the last one is whether association is analogous to a proven causal relationship. Now, this just means whether the association is similar to an exposure or a mechanism that's already well established, or that's an already known association due to previous studies. So a simple example of that is when they were looking at cigars and whether cigars cause lung cancer. Because they already had evidence from cigarettes, which is a very similar analogous kind of activity or behavior, it was more easy for them to establish that, yes, cigars are also kind of under that umbrella that are likely to cause lung cancer. So why did we talk about this? Well, in assignment 1, you'll be applying these criteria. So that's why we went through all 9 of them in such big detail. Essentially, if you follow along with the reading that was assigned for this week, it should be really, really clear how to go about doing this. You really just kind of follow the step-by-step guide. Your TAs will also be talking about this in the tutorials this week. So hopefully after this week, you'll be sick of hearing about the Bradford-Hill criteria, but it'll hopefully make a lot of sense to you. Now, for assignment 1, basically, you need to apply those criteria when you're looking at 4 studies that are exploring the relationship between exposure to radio frequency waves from cell towers and cancer. Now, full disclosure, I was inspired to do this type of assignment because of all of the, I guess, negative attention that 5G cell towers were getting this summer when I was designing this assignment. So I thought it would be kind of interesting to actually examine what the health studies say about cell towers and potential links to health. So that'll be kind of up to you to explore. Now I'll just end really quickly by saying there's a few hints about how to apply these Bradford-Hill criteria for this particular assignment. You'll notice that the studies might discuss different types of cancers, right? They're not always specific to one, and that's okay. It just might be something worth highlighting, right? That the associations might be stronger for some cancers, but weaker for others. That's perfectly fine. It's just something that you might need to draw attention to. Another kind of hint is that the associations will vary, right? They're not always going to be strong or weak. And a lot of the times, abstracts will kind of highlight the interesting results, but definitely you need to go beyond the abstract for this assignment. You'll need to read the results. You'll need to read the discussion where they interpret their findings and often give you really, really helpful clues as to whether you should pay attention to certain results in their study. Now, another quick detail I want to add before we wrap up is that you do not need to consult other sources for this assignment. You just need to read the 4 studies and you just need to read the reading about the Bradford-Hill criteria. In fact, I would say do not consult other sources than that because it might really skew your judgment about the relationship that you're supposed to look at based on only those 4 studies. So avoid it. If you want to do any sort of background research, maybe try to limit yourself and really only look at things in a very general way. Don't try to hunt for more studies to get a sense of the relationships. And finally, any limitations or uncertainties, as we kind of discussed in today's lecture, are always good to draw on for your final recommendations. So definitely keep those in mind. Now I'll end it there. So thanks so much. I know we went a couple of minutes over time here, but hopefully this all made sense to you. If you have any questions, I can stay for a couple of minutes if you want clarification on anything, but otherwise your TAs will also be talking about the assignment in this week's tutorials. So hopefully they can also answer any questions you have.